Global hunger is frequently categorized as an issue of absolute resource scarcity, but the math of modern food production points elsewhere. The true bottleneck is a failure of structural scaling and nutrient density. This graph illustrates the core problem, known as the dilution effect. As atmospheric CO2 elevates and crop growth rates accelerate, volumetric yield rises rapidly. However, nutrient density scales inversely, steadily declining as the physical plant expands. This is a highly quantifiable loss. Rapid atmospheric expansion forces a roughly 9% drop in protein concentrations across staple crops, alongside a concurrent stripping of essential minerals. Traditional extractive agrarianism, and even current vertical farms, overlook this inverse relationship. They measure success by tonnage and physical volume, ignoring the underlying chemical reality of the harvest. Solving global food production requires a pivot in how we measure yields. The agricultural sector has to stop optimizing for sheer physical volume and start engineering for molecular density. The infinite fractal descent, or IFD framework, represents a shift from traditional agriculture. Rather than treating a farm as a horizontal plot of land, IFD treats agriculture as a vertically integrated biological machine. It explicitly couples physical hardware with living wetware. To understand how this functions, we have to examine three distinct layers: the fluid dynamics governing the physical manifold, the genetic payload of the crops themselves, and the autonomous telemetry that monitors the entire stack. By synchronizing these three layers, the framework aligns maximum biological growth with the theoretical absolute minimum of resource expenditure. The foundational physical constraint of any multi-tier farm is stable fluid distribution. A tower is structurally useless if water and nutrients cannot be delivered evenly to every level. This fluid simulation demonstrates why traditional vertical farms, which rely on branching bifurcation plumbing structures, consistently fail at scale. They are extremely sensitive to microscopic manufacturing tolerances and minor port blockages. As fluid moves through these branches, pressure drops exponentially. This guarantees uneven nutrient delivery across the vertical tiers, which inevitably destroys crop consistency. Any advanced genetic or biological optimization of a plant is entirely wasted if the physical delivery mechanism feeding it cannot guarantee absolute uniformity. To solve this physical bottleneck, the IFD framework replaces standard plumbing with the recursive fractal manifold, a consecutive flow design. This animation illustrates the manifold routing nutrient solutions through nested Hilbert curve geometries. It maps a single, continuous line that extrudes into the physical manifold block. Mathematically, the filling area scales precisely based on fractal decomposition level and unit step length. By forcing fluid through this exact recursive geometry, the system maintains constant pressure. This ensures nutrient delivery with flow non-uniformity of less than 3% across all vertical tiers. These manifolds are constructed using 3D-printed high-performance peak biocomposites. By utilizing a 40% infill Hilbert pattern, the hardware saves 60% of raw material without losing structural integrity or its resistance to chemical nutrient salts. Applying this specific abstract mathematical geometry directly creates the uniform physical environment required for advanced synthetic biology. But a perfectly pressurized hardware environment requires an equally engineered biological payload. The IFD implements hybrid plant meat, or HPM synthesis. This is a dual protein intervention, utilizing CRISPR-Cas9 gene editing to actively prevent nutrient dilution within the rapidly growing crop. This diagram shows the primary step in this nutritional profiling, the cisgenic insertion of soybean LHB genes directly into the target crop's DNA. This precise insertion produces what the framework categorizes as steak plants, yields engineered to possess whole muscle iron levels and mimic the flavor profiles of traditional meat cuts. By synthesizing animal-equivalent nutrient profiles directly into plant tissues, the system entirely bypasses the massive ecological footprint associated with livestock farming. Engineering iron and flavor profiles still leaves the issue of volumetric expansion. The crop's structural carbohydrate density must keep pace with its rapid vertical growth. This molecular animation shows the required metabolic balance. The system targets the upregulation of granule-bound starch synthase 1, or GBSSI. Because it lacks a dedicated carbohydrate binding module, it interacts directly with TAPTST1 to elongate long-chain glucan polymers. Simultaneously, a secondary protein optimization utilizes mutations in the enzymes SSIA and SBIIA. This actively alters the amylose to amylopectin ratio within the plant's matrix. This precise enzymatic regulation corrects the dilution effect. It aligns volumetric expansion with structural density, guaranteeing that a larger plant is actually a more nutrient-dense plant. Managing this complex biological machinery requires real-time diagnostics. Deploying traditional, intrusive physical sensors across millions of individual plants is mechanically unfeasible and fails at vertical scale. The IFD alternative removes physical sensors entirely. It genetically engineers the crops themselves to act as non-invasive sentinel plants. The mechanism relies on an improved fungal bioluminescence pathway, known as FBP3, which is paired with a protease-responsive BRET module integrated directly into the crop's genome. Under normal conditions, the plants emit a stable yellow glow, but if subjected to a pathogen attack or lack of nutrients, the BRET module forces a spectral chill, turning the foliage a vibrant 505 nanometer green. Low-cost AI optical sensors detect this exact wavelength shift instantly. This directs the fractal manifold to autonomously adjust nutrient delivery long before physical symptoms of necrosis ever appear. Spectral telemetry effectively transforms the entire crop canopy into a unified diagnostic surface, reporting physiological health directly through light emission. When we zoom out from the individual plant level to the macro architecture of a fully operational IFD tower, these individual efficiencies compound. This cross-section shows the recursive descent cascade system. Agricultural runoff from upper donor crops is continuously repurposed to irrigate and feed the receiving crops on the lower tiers. In practice, this means the nutrient-rich runoff from the upper layers of engineered heme tomatoes perfectly calibrates the irrigation needed for receiving crops like leafy greens and mycelium below. This closed loop pushes the system to its theoretical limit, achieving a water use efficiency of 730 grams of fresh crop weight per liter of water. By optimizing nutrient metabolism at the microscale, the system enforces extreme ecological conservation at the macro scale. The final hurdle of vertical farming is rarely biological. It is the crushing reality of capital expenditure and ongoing operational costs. This flow diagram shows how the IFD framework converts precise biomass generation into a system for engineered carbon removal. By aligning precisely with the core carbon principles set by the Integrity Council for the Voluntary Carbon Market, the biomass generated qualifies as high-integrity carbon credits. These credits command prices between $170 to over $500 per ton. This high-integrity carbon revenue acts as a powerful cross-subsidy. It forces the standard $10 per kilogram minimum cost of vertical farming down, bringing the output to agricultural commodity parity. The IFD model offers a structural framework for decentralized production that addresses technical failures of scaling and nutrient density. By aligning recursive math with biological wetware, it provides a self-funding blueprint for regional food sovereignty.